Here we are going to continue our discussion of amino acid structure. However, before I jump in, I want to sort of lay the groundwork for you here. I want you to see where we're going. So what we are doing is we are building the foundation to later talk about protein structure and function. And what I mean by that is whenever you talk about a protein, it's going to have some shape. Think about this like a building, for example. When you look at a building, you can describe it based on how it looks. Is it a skyscraper or is it, a hut somewhere? Is it a cabin? When you look at a building, there is a structural description. Now, that building may have a function too. Maybe it's a car wash and it actually functionally does something. I just came up with the car wash analogy off the top of my head. I hope it works. So, when we talk about proteins, the goal is to be able to describe them with regards to their structure and their function. Now, both of those things are dependent upon a network of non-covalent interactions that stabilize that structure. As you'll see a little bit later, the interactions of protein with substrate, with enzyme with substrate, are dependent upon non-covalent interactions. And when we talk about non-covalent interactions, we're talking about hydrogen bonding, the hydrophobic effect, ionic interactions, things like that. So building towards that, this is all centered on the functional group. What functional groups are present within each amino acid, which then are incorporated into the larger polymer that is the protein. So what functional groups are present within the polymer, the full protein, that can facilitate non-covalent interactions like hydrogen bonding that stabilize overall structure. So as we go into this, what I want you to really pay attention to are the functional groups that show up. When we talk about each amino acid is unique with regards to the functional groups that are present. So please do pay attention to this. Common functional groups that you'll see in amino acids should be familiar to you from organic chemistry. You'll see here a summary of common functional groups found within amino acids. And though they may seem diverse, there are certainly some trends that you can track. So, for example, you have your alcohol-containing groups like phenol or primary alcohols, or nitrogen-containing functional groups like imidazoles, guanidiniums, amide groups, as well as carboxylates. So you'll see some common functional groups that you have probably seen before. I'm not telling you right now to go out and learn all of the functional groups that you ever saw in organic chemistry, because you don't need to know all of them, but at least have an appreciation for the general properties of, say, a carboxylate or an alcohol, a primary amine. Can they do hydrogen bonding? Are they, what is the expected charge going to be in aqueous conditions at neutral pH? And also, for those that have forgotten this at this point, we will do some review where relevant. So I'm not telling you to go relearn all of organic chemistry right now, but I just want to prepare you for a conversation that is going to center on diverse functional groups. So why do I keep harping on about functional groups? Well, what I want you to start to understand is how functional groups guide these non-covalent interactions that are necessary for substrate binding and overall protein structure. So I'm showing you a figure here that I don't expect you to understand yet. I want to sort of use this as an opportunity to introduce you to multiple things. So first, what you're seeing here is a small molecule called biotin. And here you'll see biotin bound to the active site of the biotin repressor protein. We're not going to get into what this protein does or what it looks like, really, even. That's beyond the scope of where we are at. But what I want to highlight here is that the fact that biotin is bound to this protein using multiple non-covalent interactions. Each of these dashed lines you see here represents a non-covalent interaction. Most often these are either electrostatic or hydrogen bonding type of interactions. So as an example here, this functional group that you see on the far left-hand side of biotin, this is a carboxylate. You'll see later that coloring here matters. It indicates what element is present. So red atoms represent oxygen. So here you have two oxygens. This is a carboxylate. And this is hydrogen bonded to the nitrogen group shown in blue of a primary amine. So here you have a hydrogen bonding interaction. Now, what I also want to highlight is there's another hydrogen bonding interaction on the other end here. And so this is with another oxygen with a nitrogen involved in an amide linkage. So here you have an amide group, the nitrogen's interacting with your oxygen. Before I go on and really tell you why I'm focused on this one, I just want to really highlight the result of these multiple non-covalent interactions is to position the substrate very precisely so that catalysis can occur. So the goal is to position the substrate directly adjacent to reactive functional groups so that you can do chemistry in the active site of the enzyme. Now, these non-covalent interactions center on the functional groups that are present in the active site. Some of them are obvious. Some of them directly interact with the substrate, but other ones, like this amide group, if we hop over here, here is that same amide. It's interacting with your oxygen. Cool. Now, this is particularly important because this amide actually sits on what would be assumed to be a flexible region of the protein. There's no general structure shown here. This is just, I'm showing you the polypeptide backbone. So you have amide, here's an alpha carbon, here's your carbonyl group, your alpha carboxylic acid connected to another amide. And so if you looked at this, you would think that this is flexible. However, this alpha carbon has a long aliphatic chain coming off of it. This is the R group, and at the end, a guanidinium group. This guanidinium group is hydrogen bonded with multiple other functional groups. So you've got carbonyl, carboxylate over here. And the effect is that these other non-covalent interactions hold this thing in place, which helps to stabilize the position of this alpha carbon and subsequently this amide group so that you can interact with your substrate. So what you have here are non-covalent interactions that are stabilizing the structure of the active site. All of this is centered on what functional groups are present. So I'm really trying to hammer this idea in that everything from protein structure to function is going to be dependent upon the functional groups that are present in the protein and where they are in three-dimensional space. So hopefully I can step off of my soapbox now and I can actually start talking about the amino acids. Here we're going to spend a lot of time talking about what the amino acids look like, what functional groups are present, and so I hope to have made this point and really motivated why we are spending time here doing this. Now, let's talk about how we name amino acids. Well, first, I have already told you that the naming is largely historical. What was the original source material from which the amino acid was identified? And that's really where the naming comes from. So remember, asparagine, for example, was identified in asparagus. Tyrosine was identified from cheese, where tyros is the Greek for cheese. So now, what this means is that we don't really use the IUPAC system quite like they do in organic. Now, this leads us to sort of a problem. We have to name them somehow, but we don't necessarily want to write out the full name every time we have to write out, say, a protein sequence. You know, if you have a protein that is 100 amino acids long, you don't want to write the full name for each protein or each amino acid in that protein. So we needed some way to reduce the number of characters that went into describing a protein. To put this into context, let me preface what I'm about to say by really saying I am not that old, but the example here is why we did this. So once upon a time, when you wrote computer programs, you would actually use these punch cards that you're seeing here. So this is a punch card, very much like a Scantron that you would know now, where digits have been punched out, literally punched out. And so you do this across a number of punch cards, and now here you have an actual computer program. So instead of sitting in front of a browser or a some sort of text program, typing out my computer code, I would do this on a series of punch cards and take those and insert them into a computer that would read them and then operate my program based off of the input that I've given. Okay, so now getting back to the amino acids. If everything is about the number of characters in early computing, I need to find a way to express sequence information, what amino acids are present in a particular protein. If I'm doing calculations based on that sequence, what amino acids are present, and I need to do this with the fewest characters possible. So the original system was a 3-letter code, and this was fairly straightforward. So glycine was expressed as GLY, gly, serine, ser, tryptophan, TRP, tyrosine, TYR, and so on. So the 3-letter code was fairly straightforward. You could figure out the identity of your amino acid with ease without really needing to memorize anything. However, going back to the example of the 100 amino acid protein, 100 amino acids times 3 characters. That's 300 characters. So that's still not as efficient as it could be. So what do I do? Well, I take my 100 amino acids and I find some way to express the identity of each amino acid using one character, one letter. So what we did was we designed a 1-letter code system, and it was meant to be easily memorized, which is still true. It is something that is going to have to be memorized. Now, the way that this system was designed, it is supposed to be largely based in phonetics, and you'll see how this becomes somewhat ridiculous as you go on, and I'll point that out when we get there. First, the 6 amino acids, starting with the letter, the first letter of the amino acid. There are 6 amino acids that show up more commonly than others, and we can just use the first letter in those amino acids. So C is cysteine, H is histidine, I is isoleucine, M is methionine, S is serine, V is valine. And as it turns out, there are no other amino acids that start with those letters. So those 6 amino acids, we can just use the first letter of the amino acid, and there's no redundancy. We don't have to worry about any ambiguity of having 2 amino acids that both start with the same letter. Now, this is not universally true of all of the amino acids. There are 4 or 5 others where the first letter is not unique. So for example, alanine. Alanine starts with an A. Arginine also starts with an A. So what do we do there? Well, what we do is we look at the amino acid that is more common. So alanine is far more common than arginine. So A here equals alanine. The G, in reality, you have glycine as well as glutamine. Glycine is more common, so G stands for glycine. Similarly, L is for leucine, P is for proline, T is for threonine. So these represent the amino acids. These are the first letters of the name of those amino acids, and they happen to be the more common amino acids. The next 4 in this bullet point, the The letter is meant to be phonetically suggestive. So arginine. Arginine starts with an A, but when you phonetically say it, the R kind of takes over. So when we name out arginine with using the one-letter system, we write that as R. Phenylalanine. It actually starts with a PH. This is a PHE, the phenylalanine. So phenylalanine is F. We reach a certain point here where the phonetics might be a stretch. Tyrosine is written as Y. So the thought here is that the Y sound sort of is dominant when you pronounce the word tyrosine. And the last one in this grouping, tryptophan, I'm only going to say this once, but basically if you have a lisp, you might say tryptophan. And so tryptophan is written as W. So sometimes we have to get a little bit more creative in how we go about assigning these one-letter codes. Tryptophan is an obvious example of that. So for the others are still somewhat phonetic. There are 4 others that are assigned based on letters found within their names, or suggested phonetically by their names. Aspartic acid. This is actually written as aspartic with a T, but it is named with a D based on the phonetics here. Asparagine is N. Glutamic acid is E. Glutamine is Q. So beyond a certain point here, I hope that you can see it's really going to be something of a memorization game. So you're going to need to know the one letters, and you're going to need to sort of understand why they are what they are as well. And so the last one left here was lysine. So that's just K. So this whole naming system, it'll be summarized for you a little bit later, but I want you to understand that for some of the amino acids, the one-letter code is fairly straightforward. It's the first letter or something that is phonetically suggestive, but not always. So there is some memorization here. And so to use this short little tetrapeptide here as an example, if you look at the structure here, if you know something about the functional groups and the structure of the amino acids, you can figure out the sequence. But that's a lot of characters. That's a lot of space. So we could write out the 3-letter names. This first amino acid is alanine, and if you follow through from end to end, you'll see here's your first alpha carbon. Here is a peptide bond right here. We move over. Here's our next alpha carbon. If we move over, here's another peptide bond, another alpha carbon, another peptide bond here, and our last alpha carbon right here. So if you want to know what the sequence of the protein is, you look at the R groups. Here's your alpha carbon. Here's your first R group. This is a methyl group. As you'll see in a moment, this corresponds to alanine. The next one is a phenol side chain, a phenol R group. This is tyrosine, and the next is a carboxylate that is connected by one methylene group. This is aspartic acid, and then finally glycine. So I can write this 4 amino acid peptide sequence out using 12 characters, 3 for each amino acid, ALA, TYR, ASP, GLY, or I can write this out using 4 characters. 4 characters is much more efficient than writing the 3-character naming or even drawing the structure out. If I can write 4 characters and convey all of the information that I need to you, that is much more efficient than doing the other strategies. So to put this into perspective, just to really give you that information, A, Y, D, G. That is the sequence here. So if I write A, Y, D, G, that's all I need to give to really tell you everything that you need to know. Okay, at first this table may seem a bit daunting. This is a summary of everything you need to know about all 20 amino acids. You'll see First, that the amino acids are grouped into 5 main groups that are based on the properties of the R groups. So for those that have already forgotten, everything that we talked about in part 1 of chapter 3, this is the general structure of an amino acid. At the center you have an alpha carbon. This is, with the exception of glycine, typically going to be your chiral center for that amino acid. You have an alpha carboxylic acid, alpha amino group, a proton, and your R group. So the grouping here is centered on the identity of that R group. Now, you'll find that amongst the R groups, there is a tremendous diversity. You'll find everything from nonpolar aliphatic R groups that are summarized in this first grouping. You'll find aromatic groups. You'll find polar uncharged, polar charged, whether they be positively charged or negatively charged. So the grouping here, and we're going to talk about each grouping in a moment, the grouping here is centered really on the properties of the R group. The polarity of the R groups varies widely. Some are nonpolar and hydrophobic, some are highly polar and hydrophilic, and then you're going to find that there are a few amino acids that do not fit perfectly into any one group. Cysteine is an example of this. Now, if you look at this table, you'll see far left-hand side you have the names of your amino acids. So I would become familiar with that. In the next column, you have the abbreviations. So you've got your 3-letter code next to your 1-letter code. And you'll find often that I'm going to communicate with you using the 1-letter code. It's just more efficient. This is also how biochemists tend to write out protein sequences and polypeptide sequences. So you really need to know the 1-letter code. You'll find molecular weights, which we are not really going to talk nearly as much about. You'll also find pKa values. So these are acid dissociation constants. You'll find a column for the alpha carboxylic acid group, a column for the alpha amino group, as well as a column for your side chain. Now, you're looking at this probably thinking, oh no, I have to know every single number on this table. I can't do that. Well, no, you really don't have to know every number. What I want you to see here first, I don't really care if you know the molecular weight. It's helpful to know when some amino acids are bigger than others. What's more important is to know how they are going to chemically behave. So first, when we talk about an amino acid, is it nonpolar? Is it polar? Is it charged? Is it not? And then, generally speaking, I'm never going to ask you to explicitly just give me the exact number when it comes to pKa values, but you need to know the general values. So when we talk about the pKa, say for carboxylic acids, if you look all the way down this column, the numbers basically fluctuate about 2. So when I'm doing these sorts of calculations, often they're going to be back of the envelope, rough calculations. And for the alpha carboxylic acid group, I'm going to use a number of 2. When I do the same thing for the alpha amino group, if I go down this column, you'll see that most of these are in the ballpark of 9 to 10. So maybe I use a number of 9.5. That's fine. There's no need to know the exact number for each amino acid. You will notice at least one outlier here, which is proline. As you'll see in a moment, proline actually has a secondary amine at the point of the alpha amino group, so its pKa is going to be different. Now, the R group pKa's are going to be important to know, but yet again, you don't have to remember them exactly. And in fact, in a lot of cases, I will give you these numbers, so you may not actually need to know them at all, but it is helpful to have some idea of the pKa for the side chain, the R group here. You'll see some of them at biological pH really are not going to be, they're not going to deprotonate. So tyrosine, for example, it has a phenol, an alcoholic side chain. So it has a pKa, yes, it is an alcohol, and it does have an acidic proton, but it has a pKa of about 10. So biological pH, this is always going to function as the alcohol. Biological pH is pH roughly 7.4. You'll see cysteine has a pKa of about 8. So most of the time it's going to be protonated too. Then you get to lysine, arginine. These have very high pKa's. Histidine, on the other hand, has a pKa of 6. So you'll see going forward that histidine can do acid-base chemistry at biological pH very effectively, and it is because of this pKa. The carboxylates, the negatively charged R groups that have carboxylates, have very low pKa's of about 3 to 4. And so they're usually going to function as, at biological pH, you're going to find them as the negatively charged carboxylates. So having some idea of what the side chains look like is important here, because then that helps you really to predict protonation states and charges at biological pH. To give you an example here, these negatively charged R groups. First, these are negatively charged at biological pH. You'll see in a moment that aspartate and glutamate both have a carboxylic acid as a side chain. So when protonated, a carboxylic acid has a neutral charge. It is not. It may be polar, but it is not charged. It doesn't have a negative charge or a positive charge. So that's cool, but I can't really form any electrostatic interactions that way. So because of the pKa of that carboxylic acid, for the case of aspartate it's 3.65, for glutamate 4.25, at biological pH 7.4, both of those functional groups will have deprotonated and will have adopted the negatively charged carboxylate form. So at biological pH, both of these functional groups found in these side chains can form electrostatic interactions that may be necessary for protein three-dimensional structure or binding of substrate. So here, what I'm really trying to impress upon you is that the functional group is important for three-dimensional structure as well as binding of substrate, and the functional group is dictated by really what atoms are present in it, which also dictate pKa and general chemical behaviors. Okay, so the other columns here, you'll find something called the isoelectric point, which we will talk about in a moment. You do not have to know this. You should have an idea generally what we're talking about, but know what it is, not necessarily what the numbers are exactly. And then hydropathy index. This is a measure of how polar or nonpolar is the amino acid. We'll talk about that momentarily. And also occurrence in proteins. You'll find some amino acids occur with a much greater frequency than others. The least occurring amino acid is tryptophan, which occurs about 1.4% of the time. So some amino acids occur often, and when I say often, look at leucine here, 9.1%. That is the highest number here. So basically, when we talk about occurrence in proteins, we're really talking about a 1 to 10% frequency of occurrence. The first group of amino acids that we are going to talk about is going to be a grouping with nonpolar aliphatic side chains. These are important amino acids because often they are found to be clustered within the protein interior. So if you remember the hydrophobic effect from part 1 of chapter 3, you should also remember that nonpolar molecules do not, for entropic reasons, do not want to go into aqueous water. So proteins will have nonpolar amino acids present in their sequence. So usually what happens here is the protein structure is optimized to minimize interaction of nonpolar stuff with aqueous water. So usually you'll find proteins have a hydrophobic core that is not accessible to water, that contains really most of the amino acids that we're going to talk about here. Okay, So now that I have said that, let's actually look at the amino acids themselves. So there's one observation that I have to point out first, and it is that there are two outliers here. There's glycine and there's methionine. Now, we're going to come back around to why they are outliers, but I just want you at this point to know that we do have something of two outliers here. So first, when you look at this series of amino acids, you'll see glycine, alanine, proline, valine, leucine, isoleucine, and methionine. They are ordered this way on purpose. So the R group of glycine is the smallest in this grouping. It is just a proton. So glycine has the title of being the only amino acid that is not chiral. Here's your central alpha carbon. You've got two protons, so this carbon is not chiral. All of the other amino acids are chiral. Now, alanine has a methyl group, so we get an R group that is slightly larger than just a proton. The side chain of proline is a pyrrolidine group. Proline is the only amino acid that has a secondary amine in its side chain. Then we have valine, which is a propyl group. Leucine and isoleucine are butyl groups. So remember here, leucine and isoleucine, the only difference is that basically what I've done is taken one of these carbons on the end and tacked it onto this carbon over here. So that you do that and you have isoleucine. So these are both butyl groups. Now when I look at methionine, methionine has a thioether side chain, but the thioether side chain actually resembles an N-butyl group. Carbon and sulfur have nearly equivalent electronegativities, so here this sulfur more or less could be interchangeable with a carbon, and sulfur is also about the size of a methylene group. So for that reason, we can more or less treat the side chain of methionine as an N-butyl group. So if you look at the series, you see the side chain gets larger with each next position. Okay, now let's get back to the outliers. So glycine. If you look at glycine, you're probably going to ask yourself, why is this included here? It's not incredibly polar, or it's not really incredibly nonpolar either. Glycine it has the simplest structure. It is most easily grouped here, though, just on the basis of size, but it really makes no contribution to the interactions that are driven by the hydrophobic effect. So it's grouped in here just because there's not really a good home otherwise for it. Methionine, on the other hand, it is grouped because of size largely also. Size and really the chemical behavior of the side chain. This thioether side chain is quite similar to an N-butyl group, and so that's why it gets thrown in here. Now, to point at proline, proline is different from the others as well in that it has a secondary amine as a side chain. But proline also, it does have an aliphatic side chain with a distinctive cyclic structure. The secondary amino group of proline is actually held in a rigid conformation that does reduce the structural flexibility of polypeptide regions that have proline. So you'll see as we go forward that proline has very unique properties, but it does have an aliphatic side chain. So that's why it gets included here. Okay, so these are the nonpolar aliphatic amino acids. The next group are the aromatic R groups. So a point of confusion that commonly arises in this class, and this is especially true for students that are concurrently taking Biochemistry 1 and Cellular and Molecular Biology at the same time, a point of confusion that arises for those students is the treatment of the aromatic amino acids. In the textbook that is used for cellular and molecular biology, you'll find that there is no grouping for the aromatic amino acids. They are sort of scattered into other groups based on their properties. So your textbook that we use in this class treats the aromatics as being a defined standalone group, hence us having 5 amino acid groupings, not 4. Okay, so the aromatic amino acids. First, they are all aromatic. For that reason, all of the side chains are aromatic, and for that reason, they tend to be relatively nonpolar and do contribute to the hydrophobic effect. You'll see phenylalanine is fairly hydrophobic, tryptophan also fairly hydrophobic. Tyrosine, though, it has a hydroxyl in its side chain, and it can form hydrogen bonds, and it is also an important functional group found in the active sites of many enzymes. Tyrosine is often a point that allows for easy regulation of a protein by modification of this hydroxyl group in some way or another. So you'll see tyrosine show up in biochemistry too if you take it. Now, tyrosine and tryptophan are significantly more polar than phenylalanine because tyrosine can hydrogen bond through the hydroxyl group shown here, and the nitrogen of your indole side chain of tryptophan can also hydrogen bond. So these are important properties of tyrosine and tryptophan, the ability to form hydrogen bonds. Now, before we move on, I just want to go ahead and define the side chains here. Phenylalanine has a phenyl side chain. Tyrosine has a phenol side chain. A phenyl plus an alcohol equals a phenol. And the side chain of tryptophan is the indole group. Again, all of these are aromatic and do absorb light. The light absorption properties of the aromatics is a very important property to the biochemist. The reason for this is the aromatic residues actually give the biochemist a way to quantify the concentration of a protein in solution. Okay, so let me tell you a little bit more about this. Well, tryptophan, tyrosine, phenylalanine all absorb light in the ultraviolet range of the electromagnetic spectrum. So we're talking really primarily about the range of about 230 nanometers up to about 300 nanometers. If you look at a plot of the absorbance as a function of wavelength, you're going to see that tryptophan and tyrosine absorb more closely to about 280 nanometers. Tyrosine actually absorbs maximally at 278 nanometers. Tryptophan absorbs ideally at 280 nanometers. And also there's a slight shoulder where you do get some absorption closer to 290 nanometers. Phenylalanine, on the other hand, absorbs very weakly at about 255 nanometers. So at 280, phenylalanine really doesn't absorb a whole lot. And you should notice here that the intensities are very different. Phenylalanine absorbs very poorly. Tyrosine absorbs quite a bit better than phenylalanine, but not quite as well as tryptophan. Remember, absorbance here is a metric for how well your chromophore absorbs light. These aromatics, because they are absorbing light, they are considered to be chromophores. Okay, so when I say absorb light, and what do I really mean? What are we talking about here? Well, in general chemistry and perhaps in organic chemistry, at some point you probably did a version of this experiment. Imagine you have a cuvette. This is just a glass container or a quartz container that has a solution of your molecule present. I have a lamp of some sort. This lamp emits polychromatic light, so this could be a candle, could be a light bulb, doesn't really matter. It's just shining polychromatic light. In front of that, I have a monochromator that selects for a wavelength that I'm interested in. So if I want to see absorption at 280 nanometers, it's going to pick out 280 nanometers and only let that wavelength of light through. Okay, so that specific wavelength of light is going to enter into the cuvette, and my molecule that is present in the cuvette is either going to absorb that light or not. If light is absorbed, then the intensity of light coming out on the other side goes down. So my absorbance actually is calculated as being a higher number. Okay, so this light coming out on the other side is quantified by a detector, and this is your basic experimental design for a spectroscopy experiment looking at absorbance. Now, there's another way to quantify this, and it's based on experiments like this. Here, we can look at the molar absorptivity constant, or the extinction coefficient, depending upon which language you would like to use. We can calculate the extinction coefficient for a protein based on the number of tyrosine residues and tryptophan residues that are present in that protein. At 280 nanometers, tryptophan has a defined extinction coefficient, and at 280 nanometers, tyrosine also has a defined extinction coefficient. Remember here, the extinction coefficient, all this is the probability of absorbing a photon of light at a given wavelength. So if you look at the plot of absorbance over here, at 280 nanometers, tyrosine has a defined likelihood of absorbing a photon. Tryptophan also has a defined likelihood that is higher for absorbing a photon at 280 nanometers of light. And remember that higher number, that is higher relative to tyrosine. Phenylalanine has a nearly zero extinction coefficient at 280 nanometers. It doesn't really absorb light. It doesn't have a high likelihood of absorbing a photon of light at 280 nanometers. So you can quantify, if you calculate, this is a known property, you can quantify based on the known extinction coefficient for tryptophan and the known extinction coefficient for tyrosine. These are numbers that are determined for the free amino acids in solution. So if I have a protein that has, say, 2 tryptophans and 2 tyrosines, what I would do is multiply 5500 by 2 plus 1490 times 2. So that would give me the extinction coefficient for my whole protein. It's very straightforward math if you know the individual extinction coefficients. So before I move on, I just want to really highlight the aromatic residues absorb ultraviolet light. Tryptophan and tyrosine absorb primarily at 280 nanometers. That is, they have higher extinction coefficients, or let's just say more generally, a higher likelihood of absorbing a photon that has the energy that corresponds to 280 nanometers. So they are more likely to absorb a photon of light at 280 nanometers than phenylalanine will. Okay, so this is important, and the reason I'm really dwelling on this is I want you to understand that the chromophoric behavior of proteins is important not only for the biochemist in the lab that is interested in quantifying how much protein he or she has, but you'll see systems, if you go on and take biochemistry 2, you'll see systems where proteins actually absorb light and then pass off the energy that has been absorbed for some other purpose. And a very obvious example of this is in photosynthesis. There, light is absorbed, and that energy that has been absorbed is passed off through what is effectively an electron transport chain to drive other things. So the absorbance properties of proteins can be very useful. The next group are the polar uncharged R groups. The R groups of these amino acids are more soluble in water or more hydrophilic, depending upon what language you would like to use, than those of the non-polar amino acids because they contain functional groups that can form hydrogen bonds with water. So here we are looking at serine, threonine, cysteine, asparagine, and glutamine. So you'll see here that all of these functional groups can form hydrogen bonds with water. In the case of serine and threonine, you have a primary alcohol, and then in the case of threonine, a secondary alcohol. Whether you are looking at primary or secondary alcohol, either of these can form hydrogen bonds with water. In the case of asparagine and glutamine, you have an amide functional group, and in both cases you can also form hydrogen bonds with water. The only difference between asparagine and glutamine is that glutamine has an additional methylene group that makes the side chain a little bit longer. Now, one thing to point out here is more historical in nature. Sometimes if you look back at old papers where they are interested in the amino acid composition of a protein, often you would find that researchers could not differentiate between, say, aspartic acid and asparagine or glutamic acid and glutamine. So the reason for this is related to the methods. In those very early studies, the protein was hydrolyzed all the way down to its constituent amino acids by either acid or base treatment. Now the problem there is that those are also conditions that easily convert the amide to the corresponding carboxylic acid. So sometimes if you're looking back at old studies that report the amino acid composition for proteins, the asparagine, basically the amides, asparagine and glutamine, are sort of lumped in together with the carboxylic acids, aspartic acid and glutamic acid, because you could not actually resolve the amide from the carboxylic acid. So for reasons associated with the method, we couldn't differentiate one from the other. Okay, the last member of this family, of this group here, sorry, I have allergies. The last member of this group here is cysteine. Cysteine is an outlier here because its polarity, which is contributed by its sulfhydryl side chain, so this is a sulfhydryl group. If you look at this, this will look a lot like an alcohol, just now you have a sulfur in place of the oxygen. Its polarity is contributed by its sulfhydryl group, but that polarity is quite modest. Cysteine is a weak acid and can make weak hydrogen bonds with oxygen or nitrogen. Cysteine is readily oxidized to form a covalently linked dimeric amino acid called cysteine. So all cysteine is two cysteine molecules that are joined together by something called a disulfide bond. These disulfide linked residues are strongly hydrophobic. They're very nonpolar. And so first, before I really get to talk more about that, let me look at this figure. So here you have two cysteine amino acids. So here, one cysteine, if you look here, you'll find your alpha carbon right here, your sulfhydryl side chain here. The other amino acid, another cysteine, has alpha carbon here, sulfhydryl group here. So basically you have these two sulfhydryl groups that are pointing at one another. Now upon oxidation, you're going to kick off two electrons. So basically you're going to kick off something that is equivalent to a hydride to form the cysteine. So here these cysteines become covalently linked to form the disulfide bridge or disulfide bond. Now this disulfide linkage is strongly hydrophobic. So when you find it, you're most often going to find it on the protein interior. The other scenario where you might find a disulfide bridge is in extracellular environments. So extracellularly, you have a much more oxidizing environment than you do intracellularly. So proteins that are secreted, that are extracellular proteins, are often found to have significantly more disulfide bonds than the corresponding intracellular protein. Now, why is that? Well, this is because cysteines or disulfides contribute significantly to the structural properties of many proteins. As you'll see a little bit later, one of the last things that occurs in the pathways that correspond to basically that are involved in forming protein three-dimensional structure is cysteine or disulfide formation. They help to basically sort of lock in the structure of your protein. So if you have more disulfides, you can effectively stabilize the three-dimensional structure of a protein. So disulfides are very important for a number of reasons, but the most common one is really structural support for protein's three-dimensional structure. The most hydrophilic R groups are going to be those that are either positively or negatively charged. So this brings us to our last two groupings. The first is going to be the positively charged R groups. That's what we're about to talk about on the current slide. And then in the next slide, we will talk about the negatively charged R groups. So first, I want to throw out some language. On the current slide, you're going to hear me say, you're going to hear me refer to these positively charged R groups as being basic. This is a common reference to the fact that these amino acids all have side chain pKa's that correspond to basic pH values. So lysine has a pKa that's about 10.5, arginine has a side chain pKa that's about 12.5, and histine is kind of an outlier. It has a side chain pKa that is approximately 6. However, it is still a nitrogen-containing cationic, when protonated, cationic side chain. Okay, so the amino acids that have significant positive charge at pH 7 represent these three residues. So lysine, shown here on the far left, has a primary amino group at the epsilon position of this aliphatic side chain. You'll see 1, 2, 3, 4 methylene groups and then a primary amine. Now remember, I called this the epsilon position. Alpha carbon, beta carbon, gamma carbon, delta carbon, epsilon carbon. So my primary amine here is attached to my epsilon carbon. Okay. Arginine, this is the next one. Arginine has a positively charged guanidinium group. That's what this is here. You have this functional group is the guanidinium group. Once again, let me use some of the alpha-beta language. Alpha carbon, beta carbon, gamma, delta. So my guanidinium group is attached to my delta carbon. Okay, so the last one here is histidine. Histidine also has an aromatic side chain. So this is aromatic, but this is the imidazole side chain. So histidine will absorb light due to its aromaticity, but histidine is different in that its pKa is 6. So at pH 7, you're going to have a population of histidine that is positively charged because it is protonated, but you're also going to have a population that is neutral as shown here. So what you're looking at, one thing I haven't told you is that the protonation states that are shown for each of these represent the dominant states at biological pH. So you're going to have for histine more neutral histine than you are going to have cationic histine. Lysine and arginine, on the other hand, because of their high side chain pKa's, are going to primarily be cationic at pH 7. Now, for histidine, as the only common amino acid having an ionizable side chain with a pKa near neutrality, histidine may be positively charged in that it is protonated or uncharged at pH 7. For this reason, histidine residues facilitate many enzyme-catalyzed reactions by serving as proton donors or acceptors. So histidine is very commonly observed doing acid-base chemistry. Our last grouping of amino acids are the negatively charged or acidic R groups. Once again, we see this acidic as in contrast to the basic language show up, where previously I referred to the positively charged R groups as being basic because all of the R groups have pKa's that are in the basic pH range. Here we have acidic R groups because they have pKa's that show up in the acidic pH range. So the two residues here that we're talking about are aspartate and glutamate, also known as aspartic acid and glutamic acid when protonated. You'll find that each of these R groups has a carboxylic acid in the side chain that can be deprotonated to the carboxylate, which is what is shown to you here. The difference between aspartate and glutamate is one methylene group. You'll see aspartate has one methylene group here, separating the alpha carbon from the carboxylate functional group. Glutamate has two methylene groups. So you just have one additional methylene group in the case of glutamate here. Now the pKa values are slightly different here. Aspartate, this carboxylate, has a pKa of 3.65, and for glutamate, 4.25. For reasons that we will talk about soon, this difference in pKa is chemically explainable. What you're going to see, and I'm going to give away the punchline here, what you're going to see is that the presence of the carboxylate over here, as well as the primary amine here, the alpha amino and alpha carboxylate groups that are directly connected to the alpha carbon, they are going to influence the pKa of your second carboxylic acid, whether it be an aspartate or glutamate. So in glutamate, that carboxylic acid is a little bit further away from these other two functional groups, and so you see an elevation in pKa for that reason. Now, we will get into this in more detail a little bit later. I just wanted to go ahead and introduce you to this idea. But these are the acidic residues. And here, remember, when you're going about learning the amino acids, the difference between these two acidic residues and, say, asparagine and glutamine is simply swapping out the carboxylic acid for an amide. So structurally, they are not that dissimilar. Early on in this lecture, I told you that protein structure and function is dictated by the functional groups that are present within a protein. Now you should have an understanding a little bit more broadly that this relates more to the amino acids that are present within the sequence of a protein. But, what this really is a functional group centric conversation. The choice of one amino acid versus another really gets down to what functional groups are present in the side chain for one versus another. So if you want to think about evolution more or less as nature reaching into a toolbox and pulling out different functional groups and trying out different combinations of functional groups, that is a perfectly fine way to look at this. Really, what we're talking about is a defined set of chemistries that are really guided by the functional groups that are present in those standard 20 amino acids. Now, the standard 20, you have functional groups such as carboxylic acids, primary amines, imidazoles, things like that do contribute to a lot of chemistry, yes, but sometimes it's not enough. Sometimes we need to make modifications to amino acids or use just different amino acids that confer unique chemical reactivities because of novel functional groups. So in addition to the 20 common amino acids, proteins may contain residues that are created by modification of common residues already incorporated into a protein. And this is something that is called post-translational modification, or in some cases, post-synthetic modification. So these include things like 4-hydroxyproline and 5-hydroxyproline, methyl lysine, carboxyglutamate. These are going to be important for function. And so one very, very important example here is, I'll draw your attention to gamma carboxyglutamate. This is an amino acid that is found in various blood clotting proteins, with one example being prothrombin, and in other proteins that bind calcium. In the case of blood clotting proteins, carboxylation in this way, basically adding in another carboxylate group to the carboxylic acid-based side chain of glutamate, this is important because what actually happens is you start introducing a lot of negative charge into local positions in your protein, and this will drive a change in the shape of the protein and actually facilitate binding of certain metals that ultimately lead to activation of the protein in the blood clotting cascade. So carboxyglutamation, or rather modification of glutamate residues in blood clotting proteins, modification with by carboxylation, can drive activation of proteins that are critical for blood clotting. And for those that are interested, if you dig into the science, you'll actually find that carboxyglutamate or carboxyglutamation of blood clotting proteins. This is a vitamin K dependent process, and I encourage you to go read about the vitamin K cycle. Now, there are other examples. Selenocysteine is a very good example. Pyrrolysine is another example too, but here we're focusing on selenocysteine. So selenocysteine is not a post-synthetic modification. Selenocysteine is actually, it's a rare amino acid that is not created through post-synthetic modification. Instead, it is introduced during protein synthesis using an unusual adaptation of the genetic code. So there is actually a codon that corresponds to selenocysteine. Now, selenocysteine is very important because it is a very good nucleophile. In fact, it is referred to often as nature's most potent nucleophile. If you look at it, you'll see here you have your alpha carbon, alpha amino group, alpha carboxylic acid, and the side chain here is defined by a methylene group and then a selenium atom that is bound to a proton. This looks a lot like an alcohol or a sulfhydryl. So the difference, however, is that the selenium side chain here has a pKa of about 5.4. So at biological pH, you actually have the selenium anion. Okay, why do I care? Okay, the reason I care is because when deprotonated, the selenium here acts as a very potent nucleophile. So if you absolutely need to do some nucleophilic chemistry, and you need to do so at a biological pH, say pH 6.5 or 7, serine and cysteine are not going to be able to do this because they are going to be protonated. They're going to be either an alcohol, a fully protonated alcohol, or a sulfhydryl. Selenocysteine, on the other hand, because it has a pKa that is lower than biological pH, it is going to be deprotonated. It's going to exist as an anion that is going to be able to do very potent nucleophilic chemistry. So the use of selenium here is important for facilitation of nucleophilic chemistry. Amino acids also often show up free in solution, that is, unincorporated into proteins. In these cases, these free amino acids can be metabolically active or functional as in signal transmission. So the examples that I'm showing you here are going to be examples you have probably heard of before. For example, gamma aminobutyric acid, or GABA. You probably have heard of GABA receptors, or histamine, or dopamine. You've probably heard of these before. But what I want you to see here is that they are derived from amino acids, but in their active forms, they look a little bit different. But you can still see how they might have started from an amino acid as the precursor. So in the case of GABA, you'll see you have your alpha carbon here, and then two methylene groups, and then a primary amine. So structurally, this is very similar to glutamic acid. You have your alpha carbon, your alpha carboxylate. It has the alpha amino group has been removed, but you still have your two methylene groups leading into something else. So this might have started as glutamic acid. Histamine. Here's your imidazole side chain. So here, you can see the side chain being what's left over of histidine. Dopamine looks a lot like tyrosine. You've got your phenol side chain here. Then you've got things like thyroxone that are a little bit more complicated, but you can still see the phenol here, and you can sort of see how these might have come to be. But the point here is to show you that amino acids, they may be directly incorporated into proteins, and their function unmodified, or perhaps upon incorporation into protein, there's a post-translational modification that changes the chemistry of the amino acids functional groups, or outside of the context of a protein. Maybe free amino acids in solution can be modified and be functional in regulation of metabolism or signal transmission. Some amino acid residues in a protein may be modified transiently in order to alter the protein's function.
the addition of phosphoryl groups or methyl groups, acetyl groups, adenylyl, ADP ribosyl, or other groups to a particular amino acid can increase or decrease a protein's activity. So as an example, these top three examples that are shown to you, phosphoserine, phosphothreonine, phosphotyrosine. Serine, threonine, and tyrosine, these are all alcohol-containing amino acids. The R groups all contain alcohols, whether it be a primary alcohol in the case of serine, a secondary alcohol in the case of threonine, or the phenol that is present in tyrosine. In any of these cases, phosphoryl groups can be enzymatically added on to modify these amino acids. So this is actually quite important in terms of regulation of protein activity. Sometimes what you'll find is that the addition of the phosphoryl group, so here notice the phosphoryl group is negatively charged. The introduction of the phosphoryl group introduces negative charge that may alter the local non-covalent interaction. So if I introduce a negative charge next to a negative charge, maybe the protein changes shape to alleviate that repulsive interaction. Or maybe if I introduce a negative charge via phosphoryl group next to, say, a primary amine, maybe I form an electrostatic interaction to stabilize a certain three-dimensional structure. So these modifications can lead to the change in shape in three-dimensional structure of your protein, which can also change the activity, the catalytic function of your enzyme. So there are other examples like methylation of arginine. So here I have an arginine amino acid. I have my guanidinium side chain. I've tacked on a methyl group, and this changes some of the properties, or acetylation, or methylation of other things. Or here I've got an adenylyl group added on. So the point here is just to show you that there are ways to transiently alter a protein's function by way of these post-translational modifications. I make these changes. You can think about this kind of like a light switch. I introduce some sort of change to an amino acid's side chain. That changes the functional properties of my protein, but I can also remove these functional groups as well to return the protein's activity to its original state. So this is very much a transient way to control protein function. Up to this point, we have really just focused on the modification of isolated amino acids, whether in the context of a full protein or in isolation. We've really only talked about, through co-translational and post-translational modifications, the modification of only single amino acids. We haven't talked about cases where you actually have multiple amino acids that are being modified. One very good example of that is green fluorescent protein, and this is true more broadly of fluorescent proteins, but we're going to focus on green fluorescent protein. So this is a protein that you might have come across in a biology class when talking about, say, fluorescent microscopy, where you can make these genetic fusions and fuse a fluorescent protein to another protein and figure out, say, where it's going inside of a cell, or figure out what it's doing. So green fluorescent protein is derived from a bioluminescent jellyfish that fluoresces green when exposed to UV or blue light. If you look at the structure, you'll see this is the structure of green fluorescent protein. And I don't expect you to know what this means just yet. But what I want you to see is that there's going to be a fluorescent functional group that sits inside in the protein interior. Now remember, you don't know this just yet, but the protein interior is going to be more nonpolar. Due to the hydrophobic effect, your nonpolar amino acids are going to be sort of crammed away in the protein interior in a way that is inaccessible to the polar water in bulk solvent. So here you have a fluorescent functional group that is presumably not accessible to water. Upon initial synthesis, green fluorescent protein is in fact not fluorescent. It is only upon adoption of its final three-dimensional structure, which I'm showing you here, that it becomes fluorescent. So what happens? Well, there are three amino acids, serine, tyrosine, and glycine, that are directly next to one another in sequence. So if I write out the sequence of the protein from end to end, you'll find a serine, a tyrosine, and a glycine directly adjacent to one another. Okay, so this is interesting because what happens is these three consecutive amino acids wind up undergoing a spontaneous cyclization reaction followed by oxidation to generate this. So here, what we have is a conjugated double bond system. You've got a double bond here, there's some pi overlap, double bond that leads into the aromatic phenol, and then we have more pi electrons. So we have a nice conjugated double bond system here. And this leads to a functional group. I'm referring to this whole thing here as being effectively a functional group because it functionally is fluorescent. But we have a functional group that occurs post-reaction that is fluorescent. And you can actually tune the color of your fluorescent protein too. If you swap out serine for something else or tyrosine for another amino acid or even the glycine, what you wind up doing is altering the system of conjugated double bonds there. So by altering the double bond system, the system of conjugation, you actually alter the photophysical properties of the protein. So if you don't believe me, let me demonstrate this to you. So here, what you are looking at is that exact thing, the modification of the local environment around that conjugated pi system, which effectively, I can't use PowerPoint, effectively changes the color of your protein. So you can modify anywhere from blue to green to yellow, orange, all the way down to red. And you can see that the design, this is an example of protein design, the design and engineering of that conjugated pi system directly impacts the photophysical properties of the protein. Now there are uses for these fluorescent proteins. You can actually make these genetic fusions where you fuse genetically the amino acid sequence for a fluorescent protein to, say, a protein that you're interested in. And then you can do fluorescent imaging and you can figure out where in an organism your protein winds up. Say this is a protein that you're interested in and you did imaging, you'd see, oh, it's actually here in the organism, not down here. There's actually something else that's purple down here. And of course, you would know what was purple based on your experimental design. So this is a very useful thing to be aware of. The engineering of these fluorescent proteins, it is an example of a post-translational modification because the protein is not fluorescent upon the initial synthesis in the cell. So it only becomes fluorescent later. Hi, now you can finally see me. So my goal for this lecture has been to introduce you qualitatively to amino acid structure and really to get you to start thinking about how amino acids might behave when incorporated into a protein structure. What I want you to really think about here is how the amino acids are different relative to one another. What makes them different? Well, the answer to that is the functional group. They are all defined based on different functional groups that show up in their side chains, their R groups. So this is really important because if you understand how the functional groups are generally going to behave, what makes a primary amine different from, say, a carboxylate? If you understand the difference here, you can make some pretty major predictions about how an amino acid is going to impact both structure and function for a fully structured and functional protein. So that's really one of the goals here. So next time, we're going to start talking about some of the quantitative aspects of amino acid side chain behavior. When we talk about pKa, or at least when we have talked about pKa thus far, it has largely been qualitative. They are going to be protonated or not protonated at this pH, and it's really sort of been, we talked about it at a distance. Next time, we are going to really dive in. We're going to spend a lot more time talking about this in depth. So get ready for that. Um, Once we finish the next lecture, we can then move into a discussion of proteins from a functional perspective. We can talk about, the full length protein as a polymer. How does it behave? The behavior of the full protein as a polymer. That's going to be different than the individual amino acid. So we'll spend time talking about that as well.